Did you know that up to 25% of herbal drugs tested contain traces of heavy metals and toxic substances? Yes, you heard it right. It's a sobering reality that even the most natural of remedies can host a cocktail of contaminants. We're talking about herbal drugs, those nature-derived substances that we often consider as safe alternatives to synthetic medications. But the prevalence of contaminants in these herbal products is a growing concern, and one that we need to address head-on. So what's the culprit behind this issue? Well, the answer lies in the lack of stringent quality control measures in the production and distribution of these herbal drugs. While they offer significant therapeutic benefits, it's essential to remember that the safety and efficacy of these drugs are paramount. And this is where the importance of quality control steps in. Quality control in herbal drugs is not just about ensuring the right plant species or the correct extraction method. It goes beyond that. It's about taking a closer look at the hidden dangers that may lurk within these herbal remedies. It's about testing for the presence of pesticide residues, arsenic, toxic metals, microorganisms, aflatoxins, and even radioactive contamination. These are substances that can pose serious health risks when consumed, and it's our responsibility to ensure they don't make their way into our bodies. The reality is, no matter how beneficial these herbal drugs may be, they can also be a double-edged sword. And it's a sword that swings heavily on the side of potential harm if we don't take the necessary precautions. The importance of quality control in the production and distribution of herbal drugs cannot be overstated. It's our best line of defense against these hidden dangers. With such alarming facts, how do we ensure that the herbal drugs we consume are safe? Let's delve into the world of quality control parameters. Because at the end of the day, health is wealth, and wealth without health is a bitter pill to swallow. So let's take charge of our health by understanding the hidden dangers in our herbal drugs. Scene script. Pesticide residues in herbal drugs can pose significant health risks, but how do we detect them? Well, the principle of pesticide residue determination is based on the extraction, separation, and detection of these potentially harmful substances from the herbal product. The process aims to identify and quantify the residual presence of pesticides, ensuring that the levels are within acceptable safety limits. The procedure starts with the sample preparation. The herbal product is ground into a fine powder, then subjected to extraction using a solvent. This extraction process aims to dissolve the pesticide residues present in the sample. Once the extraction is complete, the next step is the separation and detection of the pesticide residues. This is where sophisticated analytical techniques come into play, such as gas chromatography and mass spectrometry. Gas chromatography, or GC for short, is a technique that separates the different components of the sample. The extracted solution is vaporized and carried by an inert gas through a column. The different components in the sample will separate based on their volatility or their tendency to remain in the gas phase. Following the GC, the separated components are then analyzed by mass spectrometry. This technique identifies the different components based on their mass-to-charge ratio. It provides a unique fingerprint for each pesticide, allowing for their identification and quantification. So why is this important? The determination of pesticide residues is a crucial part of quality control for herbal drugs. It ensures that the products we consume are safe and free from harmful levels of pesticides. Without these checks, there's a risk that unsafe levels of pesticides could make their way into our bodies, leading to potential health risks. In conclusion, the process of pesticide residue determination is a complex but essential step in ensuring the safety and quality of herbal products. It requires sophisticated techniques and careful execution, but the end result is worth it. Safe, high-quality herbal drugs we can trust. Now that we know how to detect pesticide residues, let's talk about other harmful substances that might lurk in our herbal drugs. Arsenic and toxic metals in herbal drugs? That's a scary thought. Fortunately, science has provided us with effective techniques to detect these harmful substances. Let's delve into this a bit more. Arsenic and other toxic metals can sometimes make their way into herbal drugs during the cultivation process, through polluted soil or water. This is where the need for detection methods comes into play. One of the most commonly used techniques is atomic absorption spectroscopy, or AAS for short. The principle behind AAS is simple yet ingenious. It relies on the absorption of light by free metallic ions. In essence, a sample is vaporized, and then it's exposed to light of a specific wavelength. The amount of light absorbed is directly proportional to the concentration of the metal in the sample. Now let's talk about the process. The procedure of AAS involves several steps. First, the herbal drug sample is dried and ground into a fine powder. This powder is then digested using a strong acid to break down the organic matter and release the metals. Once the digestion is complete, the sample is diluted and aspirated into the spectrometer. The spectrometer then measures the absorption of light, allowing us to determine the concentration of the metal in the sample. The significance of this process cannot be overstated. Detecting toxic metals is crucial for ensuring the safety and efficacy of herbal drugs. By identifying and quantifying these metals, we can prevent potential health hazards and ensure that the herbal drugs we consume are of the highest quality. Keep in mind that while these procedures are highly effective, they require sophisticated equipment and trained professionals to carry them out. It's not something that can be done in a home setting, but we can all play a part by demanding transparency and quality control in the production of herbal drugs. But the list of potential contaminants doesn't end here. Let's move on to microorganisms. Scene script. Even tiny microorganisms can pose big threats when it comes to the safety of herbal drugs. When we talk about herbal drugs, we often overlook the possible presence of harmful microorganisms and aflatoxins. Yet, these microscopic entities can significantly affect the quality, safety, and efficacy of these remedies. Microorganisms are ubiquitous. They're in the air we breathe, the food we eat, and yes, even in the herbal drugs we consume. Bacteria, fungi, and yeast can all potentially contaminate herbal products. If they multiply unchecked, they can cause spoilage, alter the drug's properties, and even cause illnesses in consumers. This is where microbiological testing comes in. The principle is straightforward. Let's create conditions that allow any lurking microorganisms to grow, then identify them. The procedure involves taking a sample of the herbal drug, preparing it, and then incubating it in a culture medium. Over time, any microorganisms present will multiply, forming colonies that can be counted and identified. Now let's shift gears and talk about aflatoxins. They're a type of mycotoxin, which are toxins produced by certain fungi. Aflatoxins are infamously potent and can cause serious health issues, including liver damage and cancer. Detecting aflatoxins in herbal drugs is crucial, and one common method is immunoaffinity column testing. This test uses antibodies that bind specifically to aflatoxins, effectively trapping them. The principle is simple, but the implications are vast. Here's how it works. The herbal drug sample is prepared and passed through a column filled with these antibodies. The aflatoxins in the sample bind to the antibodies and stay in the column while everything else washes through. After that, a specific solution is used to release the aflatoxins from the antibodies, and they can then be identified and quantified. So the presence of microorganisms and aflatoxins can pose significant threats to the safety of herbal drugs. But through vigilant testing and quality control, these hazards can be identified and eliminated, ensuring the safety and efficacy of these natural remedies. And lastly, let's discuss one more potential contaminant: radioactive substances. Radioactive substances in your herbal drugs? As unlikely as it sounds, it's a possibility we can't ignore. The reality is, traces of radioactive elements can make their way into herbal drugs during cultivation, processing, or packaging. This is where quality control parameters come in, ensuring that what you consume is safe. Let's delve into the principle and procedure of detecting radioactive contamination. The star player here is gamma spectrometry, a technique that measures the energy of gamma rays emitted by radioactive substances. If a sample emits more gamma rays than what's considered safe, it's a clear sign of radioactive contamination. Now you might wonder, why is this so significant? Well, exposure to radioactive substances, even in small amounts, can lead to serious health issues over time. Therefore, detecting and eliminating radioactive contamination is a crucial step in ensuring the safety of herbal drugs. So, as we have seen, ensuring the safety of herbal drugs is a complex process involving multiple steps and techniques. In the world of herbal drugs, safety is paramount. We've journeyed through the hidden dangers that can lurk within, from pesticide residues to toxic metals like arsenic. We've seen the importance of rigorous testing for these substances, as they can pose significant health risks if left undetected. We've delved into the microscopic world of microorganisms and aflatoxins, highlighting the necessity of monitoring these elements to prevent harmful effects. We've also explored the potential for radioactive contamination, a less common but equally serious concern. Each of these quality control parameters plays a vital role in ensuring the safety of herbal drugs. They are the safeguards that protect us from unseen dangers, the checks and balances that keep us healthy while we seek to benefit from nature's bounty. So let's continue this journey of discovery together. There's so much more to learn, to understand, to explore. Remember, knowledge is power, so subscribe for more because you won't find me later.